First of all, I'd like to thanks to the organizing committee of this workshop and Rajiv Varsini for inviting me to give a talk in ICRISAT. My first visit to ICRISAT was made around 20 years ago, probably in 1995. At that time, the scientists in ICRISAT was beginning to use some marker technology by some RFLP and RAPID. However, science is changing very rapidly, and with the recent release of some genome sequence of chickpea and PGMP, the breeding program will be accelerated for these legume crops. Now I'm going to talk about another legume crop, mungbean, Vigna radiata. Today I'm going to overview the genome and the epigenome sequence of mungbean. And also, I'd like to introduce briefly the initial study of identification of QTL for major agronomic trait in mungbean. As you know, mungbean is fast-growing and warm-season legume species belonging to the papilionoid subfamily. And the mungbean, the genome structure of the mungbean is similar to the agriculturally important crop glycine max and common bean, and a little bit different from lotus and medicago model legume plant. And India has the largest production area of the mungbean in the world. And also, based on the total production of mungbean, India was the first, the second China, the third is Myanmar, and you can see a pretty large amount of production in Indonesia and Thailand and Pakistan. And however, the yield of the mungbean for India, just a national yield is about less than 0.5 metric tons per hectare. So, so maybe future breeding program for the mungbean should be directed to the increase seed yield. For genome sequencing, let me introduce the genome of the Vigna radiata. Diploid chromosome number is about 22. The genome size is about 0.55 gigabase. And for the glycine max, you know very well, about 2N is 40 and 1.12 gigabase. So the genome size of the Vigna radiata is the half of the glycine max based on the chromosome number and the genome size. And also, within Vigna species, you can see some Vigna reflexo pilosa. The chromosome number is 44, and the genome size is 0.97 gigabase. So this Vigna reflexo is a duplicated form of this Vigna radiata. So if we compare those genome sequence across three plant species, we can understand the duplication, genome duplication. And also, in addition to this Vigna radiata, we have sequenced the wild mungbean, Vigna radiata sublobota, and to understand the domestication history of the mungbean crop. For this study, we selected the VC1973A, developed at the AVRDC in Taiwan. And this VC1973A was introduced into Korea in 1982 and renamed as Sananokdu. And you can see this VC1973A were grown in many countries, and from low latitude Thailand to high latitude Canada. So probably this VC1973A is insensitive to photoperiod. For genome sequencing of VC1973A, we implemented several types of libraries for 11 high-seq, and also we used the GS flags. A total of over 360 sequencing depths were produced for this genome sequencing. And in addition to VC1973A, we also briefly sequenced the V2984, this another local cultivars of mungbean grown in Korea. So this is the parent for construction of a mapping population. So by comparing those sequences together, we can develop some SNP markers for construction of a genetic map. And also, as I mentioned earlier, we also sequenced the wild mungbean and also duplicated the Vigna reflexor B1160 also sequenced using this fragment and MatePad 5K libraries. And to know the epigenome sequence, we adopted the bisulfite sequencing technology for these two Vigna radiata. Just briefly, the genome assembly, for the genome assembly of the mungbean Vigna radiata, we have about 2,748 scaffolds, and the M50 of the scaffold is the 1.5 megabase. And based on, we ordered the scaffolds based on the markers, SMP markers in the genetic map, and the pseudo-molecular size is about 35 megabase. And also we did some kind of genome assembly for the Vigna radiata subrobota wild species, and also duplicated Vigna reflexor pilosa. And let me introduce just briefly the genome overview of the mungbean. And we complete about 431 megabase of the estimated genome size, 540. About 80% of the estimated genome size was sequenced. And out of them, about 50% consists of repetitive sequences. And we also, using some RNA sequencing analysis from 4 different tissues, we annotate about 22,427 high confidence protein coding genes. And by comparing with another genes in Oryza sativa, Arabidopsis, and the Medicago, and the Glycimex, we found Vigna radiata specific 160 genes in Vigna radiata. And as you know, the soybean Glycimex is characterized with 2 times duplication. And 58 million years ago, ancient duplication, and recent duplication 30 million years ago. For the Vigna radiata, we could not see any evidence for the recent duplications like common bean. And we saw about 59 million years ago ancient duplication like soybean. However, the duplicated Vigna reflexor is a little bit distinct from soybean, and you can see very recent duplication 6.8 million years ago. However, we see a little bit kind of ancient duplication 49 to 74 million years ago. So probably the Vigna reflexor probably have a rapid fractionation of the duplicate after ancient duplication or ancient whole genome duplications. This is in contrast with the rapid whole genome duplication in soybean crops. And we also divergence between the mungbean and the soybean were calculated to be 25.5 million years ago, and this is a little bit later than the divergence time between mungbean and the PGMP, 33.4 million years ago. You can see a large synteny of the mungbean with soybean, and of the mungbean with pigeon pea. And during speciation and domestication of Vigna species, substantial adaptation to the some kind of climate change and the cultural environment have occurred, changing morphological and physiological characteristics of Vigna species. So to understand the speciation of Vigna species, we collected about 22 accessions of including 16 Asian Vigna species, 1 African, and 1 Eurasian Vigna species. So here is the list of the some Vigna species. RNA sequencing transcriptome analysis have been done for these 22 accessions. And we found that about the 7,202 orthologous genes were found across 22 species. And based on the comparison of these genes, we can classify 3 major classes. And this is consistent with some classification by phenotypic data. And in addition to this, we found that some Vigna reflex is like allopolyploidized genome structure. So one of the parents should be Vigna 3 Norway, but we don't know the other one. But should be we found that this one, and also Vigna radiata. We calculate the divergence time between Vigna radiata and its wild type, Vigna radiata sublobota. It's about 1.20 million years ago. So domestication has been occurred around 6,000 years ago. So this species divergence predated domestication, indicating that before domestication, the Vigna radiata was existing in the nature. This is the same conclusion in soybean. So probably the nature before domestication, the Vigna radiata and Vigna radiata sublobota may existing together. And we also found some genomic difference between Vigna radiata and its wild type Vigna radiata sublobota. And the 1.8 million SNPs were found. Out of them, 23,000 SNPs are non-genomics in 9,660 genes. And we also found some small deletion and insertions, about 750 frame shift we found. And to understand whole genome methylation pattern, we did some, we used the sodium bisulfite treatment for the 2 Vigna radiata. One is the VC19738 and the other local cultivars to find some epigenetic site and some genes. And as I mentioned earlier, Soranokdu, the VC19738, and the Gyeonggijere was used for this study. Kampengsen KPS1 is another name of in Thailand, the same variety. And the 2 Vigna radiata were characterized with different, the characters of 2 varieties, you can see very variable characters here. For example, the VC1973 has higher, greater seed weight than the Gyeonggijere 5. In addition to this, you can see very difference in characters such as hypocotic color, and some immature produce, lost on seed surface, and some disease resistance for the powdery mildew. And we also, this is the methylation data between 2 genotypes. For regardless of genotypes, you can see the CD is the major methylated form, and the next is CHG, and the next is CHH. And regardless of genotype and methylation patterns, intergenic regions show the higher methylated patterns. And we compared this methylation with the other plant, Arabidopsis and the soybean, already reported in previous study. The major difference between Arabidopsis and the soybean is the methylated CHD. The frequency of the methylated CHD is the half of the methylated CD in Arabidopsis. However, for soybean, you see the frequency of the methylated CHD is not much different from methylated CG. So our two varieties, mungbean variety VC19738 and V2984, was similar to the some soybean crops. And we also locate all the methylation in mungbean. This is the one of the patterns, VC19738. You can see, for example, the gene density is pretty low. It's probably heterochromatin regions had a higher frequency of methylation. In comparison with that, the euchromatin regions with the high gene density had lower methylation patterns. And this was done by Dr. Smith, published in Genome Research just last year. One of the patterns about CG, and they identified the 10,000 pair of paralogous because of the duplication in soybean, and they checked the some methylation patterns between paralogous, and you can see here most of the recently duplicated genes show the similar methylation between two paralogous. However, the mungbean crop has the different methylation pattern with from the soybean crops. So as I mentioned earlier, this is a recently duplicated gene in soybean. So soybean, as I mentioned earlier, soybean has the two different duplication, ancient and 58 million years ago, and the recently duplication 30 million years ago. So this duplicate happened by the recent duplicate gene may have the same methylation pattern between parallels. However, ancient duplicated, you can see divergence in ancient duplicated duplicators, indicating that after the methylation pattern after divergence after ancient genome duplications diverged, so they had diverged very much compared to the recent duplication. And due to the lack of the recent duplication in mungbean, you can see very small amount of correlated genes, paralogous. Okay, we compare the FAR site between two genotypes. About 98.6% of the FARs were conserved between two genotypes. Only 1.4% show the FAR sites. So further studies are in progress to identify this type of the meaning of the FAR site in this using this data. And my talk, I will give a talk just initial study of QTL mapping. We produced a mapping population VC19738 and the 2984, we had about 190 recombinant inbred lines, and we evaluate all the major agronomic traits for these lines, flowering time, including flowering time and uniform maturity, or some kind of disease resistance. And QTL analysis have been done using these mapping populations. And for construction of a genetic map, we used the genotyping by sequencing at BGI, and we a total of 1.9, about 1,900, 2,000 SNP markers were called, and out of them, about 21 SNP markers were mapped onto the genetic map. And actually, I skipped the explain this one, and we using these genetic markers were used for ordering the scaffolds and to make about 35 of empty lengths of pseudomolecules. And using this genetic map, we identify some QTLs. And I can explain just a few of the traits here. And the days to flowering, number of branches, and plant height and brown spot. And most of the data shows some transgressive segregation in recombinant inbred lines. And this is the sum of the QTL identified in this study. Flowering time, first flower, lodging, and some kind of disease here. But for the mungbean, it is very difficult to harvest one time because some pods are already mature, but the other pod is still maintain green color. So farmers should harvest 2 or 3 times harvest. So because of that, synchronous maturing time is very important character in mungbean. So the flowering in mungbean is very important characters. So we identify some flowering QTLs. This is an example of one QTLs, and we identify one major QTL on chromosome 3. This is we compared those QTL with some QTL in soybean crops. We found that already the gene for flowering in soybean was already identified in these regions. One of those white chromes, E3 genes, and also another gene was also reported in soybean on other chromosomes. This is the recent duplicated, another ancient duplicated genes also have these genes, but one of them does not have that genes. So by comparing this data with of QTL position of mungbean with the soybean crops, we were able to find some this mapping population is pretty good for the further mapping of the agronomic trait. Okay, this is my final slides, and actually all right. Dr. Kang is the main researcher in my groups for these mungbean crops. He was in charge of bioinformatic analysis and writing this manuscript. And we have a good collaborator from the University of Georgia, Scott Jackson. Actually, when I started to sequence these genomes, we discussed about I chose the mungbean because of the Scott Jackson's advice. And some Roland for the mapping population also he provides some QTL positions of the identified in AVRDC, and we mapped those genes in our sequence data. And the Rajiv Barsini was help our some genotyping by sequencing to construct genetic mapping populations. So this is the one of bottleneck, and we finish our sequencing data to make some pseudomolecules. So great help from Rajiv Barsini. And the Pirasoxylin knives at Kasarto University, they provide some kind of his group provides some Vigna species and give a lot of information about the Vigna speciation. And we also have a collaborator from Rural Development Administration in Korea. Okay, thank you.
Keeping to time. So we have time for some questions.
Dr. Lee, I have a question, rather two if I'm allowed. Actually, I wanted to know that was there some specific criteria when you chose the parents for this sequencing initially, particularly the wild mungbean?
So you mean the crop?
Yeah, I'm
Okay.
asking that.
Actually, the mungbean was not important in Korea. So actually, however, to understand the genome duplication of soybean crops, I selected the mungbean at first. But when I did this study, I did not know the importance of the mungbean exactly. However, after sequencing the data, I'd like to use the actually to understand the genome duplication in soybean. I choose the mungbean.
Okay, so these genotypes must be having some specific traits also, which you later, during the course of study, you came to know of some specific traits in these genotypes. And my second question is, actually, when you went for duplication check between different species, did you consider some more related species like azuki bean, urd bean, or maybe physiolus? Why I'm asking is because when I am going for cross-transferability of markers, so I find it very easy to transfer markers from azuki bean, from black gram, from rice bean, and even physiolus.
Yes, actually we had some data, as you know, here we have a lot of data about transcriptome data of different species, and so we did not compare those sequence transcriptome data exactly, but actually we started the genome sequence of Vigna angularis, so some kind of initial study for that we will do soon.
Okay, thank you very much. Any other questions? Well, if not, we're just 5 minutes delay, which I think is more or less where we started. So I think I'd like to thank again all of the speakers for keeping to time and for their very interesting talks. So let's thank them all again.